This video is about gene mutations and their effects on DNA. A mutation is described as a mess-up in the DNA sequence. Before we can go over different types of mutations, we must first review how DNA is transformed into proteins. This is called the central dogma of molecular biology, which states that DNA is transcribed into RNA, which is then translated into amino acids. These amino acids then bind together to make specific proteins. An example of this would be the DNA sequence GTA, which is transcribed into the RNA sequence CAU. This specific RNA group is then translated to the amino acid HIS. Now, mutations can occur at any point of the DNA sequence. And as we see here, if one DNA sequence is mutated, it can transcribe the wrong RNA sequence, which would code for the wrong amino acid. In this video, we will be covering 4 types of gene mutations. These 4 types are Misssense, Nonsense, Silent, and Readthrough. But before I can explain each of these mutations, let me explain the difference between transition and transversion mutations. Transition mutations occur when 2 purines are switched or 2 pyrimidines are switched. An example of this is when an A is mutated to a G, or if a C is switched for a T. A transversion mutation occurs when a purine is switched for a pyrimidine, or vice versa. This would be if an A is mutated to a C or T, or if a C is mutated to a G or A. Now, onto the 4 different types of mutations. We will start with the missense mutation. As we can see, the normal DNA sequence and the mutated DNA sequence below. We can see that the C is changed to an A. This would be a transversion mutation. This mutation then changes the RNA sequence from UUG to UUU. So originally the amino acid codes for LEU, but now codes for PHE. A missense occurs when a mutation alters the coding for one amino acid to another one. Our next mutation is called the nonsense mutation, which changes a regular amino acid into a stop codon. We can see in the example that in the DNA below. The G in the DNA sequence is mutated to an A, which makes this mutation a transition. The RNA sequence is also changed from CGA to UGA. This is a stop codon which ends the replication process. Silent mutations are the next we are going to focus on. Silent mutations occur when there is a change in the DNA and RNA sequence, but there is no change in the amino acid sequence. In the example below, we can see that C is mutated for a T, making this a transition mutation. This changes the RNA sequence from UUG to UUA, but both of these code for the amino acid LEU. So there's a mutation, but no one would ever know unless they looked at the DNA. Our last mutation is a read-through mutation. This changes a stop codon to a normal coding amino acid. Here the A is turned into a T, changing the stop codon UAA to the regular amino acid coding AAA. Thanks for watching.